Hello, my name is Jesus Rodriguez. I'm the Capstone 2 lead for the Analyzing Biological Networks group for this semester, for the fall 2022 semester. Um, my team consists of Jose Carlos Luzbel, Maria Hernandez, Seyun Cho, Bin Hao Lu, Jose Altamirano, Mojir Achale, Ralph Calixte, Austin Escobar, Rebecca Gonzalez, Adio Hai, Alexis Loriga, and Daniel Milanes. Our product owner for this semester was Ananda Mondal, and our instructor was Masoud Sajjadi. So, a quick introduction. Cytoscape is an open source software platform for visualizing complex molecular interaction networks and integrating them using a variety of functionality, such as annotations and gene expression profiles, among other features. Essentially, Cytoscape is basically the software we're using to kind of use our plugin. And all Cytoscape is doing is it's kind of like the software that's allowing us to use graphs and view graphs, while Cytoscape will offer some additional functionality, as stated before, such as giving the nodes names, or being able to visualize it much more clearly. There's a lot of other functionalities, such as being able to color the nodes, or resize the nodes, etc., etc. And that's basically just the software we're using for the semester. The SciFinder is a plugin built for Cytoscape, which helps find the subgraphs of a given network using multiple different or unique algorithms. Essentially, SciFinder is the plugin that we were working on over the semester. And this plugin is something that we use in Cytoscape in order to test out a lot of, as of recently, it's been a lot of community detection algorithms, but there could be other algorithms that are used as well. But for the most part, a lot of the algorithms that we're using for the purposes of finding these subgraphs or communities, as we'll explain later, is using a lot of community detection algorithms. And we'll try to go into that a bit later. So the SciFinder plugin, as of right now, is solely focused on detecting the subgraphs of any given network, but it doesn't have any built-in functionality to perform research on said subgraphs. So essentially, as stated before, the SciFinder plugin and the algorithms that are implemented into the SciFinder plugin are used to be able to split apart the graph into different subsections depending on the communities they fall best into, which again we'll explain a bit later. But it doesn't have any built-in functionality, to perform the research on them. A lot of it, what it's done is just splitting up into communities for researchers to study it, which I'll say right now. These subgraphs of a complete biological network, such as protein-protein interaction networks, can help researchers detect disease progression at the protein network level. So essentially, by studying these subsections of the graph, partitioned by different community detection algorithms, you can kind of study them and find out differences within the communities, and find maybe interesting data, if you're lucky, or if it's possible, depending on the community detection algorithm that is being used. During our time working on the project, we've been able to complete the InfoMap algorithm and implement it into the backend of the SciFinder plugin, which is called the Subgraph Finder. So now, community detection. So community detection is just known as the process of discovering cohesive groups / clusters within a network. So as you can see in the image on the right, there are, to the naked eye, there are 4 different communities. There's the one defined by the ones in orange, the ones in light orange, blue, light blue. And we can see that give or take any of those communities, say the orange one, you see a lot of the nodes there are very clustered up, and it's very tightly knit compared to you're trying to traverse from the orange to the blue, and it's not as tightly knit, the connections there. So you see a lot of closeness, and it's basically saying this is being defined as a community to the naked eye. However, graphs that are used for testing purposes, and especially more complex ones, are not going to be so easily visible to see the communities within them, and that's why we need these complex algorithms to find these communities. So community detection actually does play a vital role in the success of the SciFinder plugin, as it's what defines what the subgraphs or the communities of a given network will look like. So like you wouldn't expect to see too much difference between like the communities generated between different community detection algorithms. However, like some of them can be. It really just depends on the ones you use. Each community detection algorithm has its own purpose and its own role to play, and picking the right ones for the purposes you are trying to look forward to um, are very important when trying to get the information you want to get. Um, and so, in a given protein-protein interaction network, such as the graphs we're testing on, these clusters can be indicative of interaction between multiple different communities detected within said network, and depends on which algorithm is employed. Again, as stated before, if you're implementing an algorithm called Luvian, which is a community detection algorithm implemented by a previous group, we could expect to see like different results than like implementing one called leading eigenvector or walk trap or Gerben-Newman. These are all just community detection algorithms that I'm listing out, but you could expect to see very like some different results between each of them. Each of them are used for their own purposes. So to kind of end the video off with a little bit of like knowledge that will be needed going forward, description length is something that is very important to what we are working with in the for this semester. So what is meant by a description length? A description length, for our purposes, just defines how many bits are needed to describe a traversal through a network. So a bit is just like, like a 1 or a 0, and it's just like saying how many, like, if we had like a traversal through a network, it like traveled like, maybe 100 nodes in a graph or something, how many bits would it take to represent the traversal? And so using that, we would want to also try to minimize the description length. And there are algorithms whose purpose is to look through any given amount of partitions within a graph and determine which partitioning would net the shortest description length. And those different partitionings are known as modules. And the algorithm, or one such algorithm that we are implementing, is known as the infomap algorithm. And essentially, the infomap algorithm, as we'll explain in like future videos, serves to kind of look through the different partitions within a graph and see which one would net you the shortest description length. And that's essentially what it's like, the whole purpose of what it's trying to do, and that's how it like kind of optimizes the community detection. Again, this will be something explained in much further detail as we go along in the videos, but that will be all for the intro video, and we'll see you guys later. Take care.